Good morning, students. In today's lecture in Transfusion Medicine subject, we are going to learn about the ABO and RH blood grouping. This lecture is presented to you by Sanjay Gandhi Postgraduate Institute of Medical Sciences, Lucknow. So the learning objectives for today's lecture are understanding the inheritance of the blood group systems, how they are passed, how the genes for the blood group systems are passed from the parents to the children, how the antigens are synthesized, what are the antibodies and how the antibodies against those antigens are formed inside the human body, what is their clinical significance, and then mainly we'll learn about the ABO and RH blood grouping. We'll also learn about the practical aspects, what problems do we face while doing the blood grouping in the blood bank and how to issue. So a brief introduction about the blood groups. What are human blood groups? So the red cells have certain antigens on their membrane surface, which can be either of carbohydrate type or glycoprotein type. These are called the antigens. ABO blood group system mainly has carbohydrate antigens, while the RH blood group system has glycoprotein antigens. These antigens are unique to the individual. They are considered as foreign if they are transfused to another individual. If those antigens come in contact with a similar affinity antibody, it leads to agglutination of those red cells. And as per ISPT, International Society of Blood Transfusion, till 2023, we have recognized 45 of such antigen systems, which are blood groups. The presence or absence of these antigens are used to classify these blood groups. Major ones are ABO and RH. Minor ones are many, for example, Kell, Kidd, and Duffy. The ABO blood group system is the most widely known and most medically important blood group. It was discovered by Karl Landsteiner in the 1900s. There are two antigens and two antibodies in this blood group system. The two antigens are A and B. The two antibodies are similarly anti-A and anti-B. This blood group has two antigens, which are namely A and B antigens, and two antibodies, which are anti-A and anti-B. The antibodies are present in plasma and the antigens are present on the red cell surface. Landsteiner, while discovering the ABO blood group system, gave a law which is known as Landsteiner's law. It says that the plasma contains natural antibodies to A or B if these antigens are absent from the red cell of that person. Now, what does that mean? This means that there is a reciprocal relationship between the ABO antigens and the antibodies. For example, if the antigen present on the red cell is A, as you can see in the first row, the antibody which would be present in the plasma or serum of that individual would be anti-B, and the blood group would be typed as A. Similarly, if the antigen that is present on the red cells of the individual is B, the individual would naturally have anti-A antibody in its plasma or serum, and the blood group would be B. Now, what do we mean by naturally occurring antibodies? These are antibodies which are present in the human body without any exposure. So, although the ABO blood group antigens are regarded as RBC antigens, they are also present on variety of other tissues in humans, as well as their similar structures with the ABO blood group system are found on other organisms in the nature. For example, some bacteria, plants, and other animals have antigenic structures which are mimicking the A or B antigen of the red cell. This leads to antibody formation in humans. So when we say naturally occurring and no exposure is required for the antibodies to form, it is partially true that when the baby, for example, when the baby is born, the babies do not have any anti-A or anti-B in their blood or in their plasma or serum, as you would say. But as the baby's gut after feeding begins to become unsterile, the baby is exposed to various bacteria and virus and other antigens from the environment. The baby starts to form anti-A and anti-B antibody due to exposure to those antigens which mimic the A or B antigen of red cells. ABO blood group system inheritance follows the basic Mendelian principle. They are co-dominant. Two genes are inherited, one from each parent. Individual who is A or B may be either homozygous or heterozygous for the antigen. For example, AO and AA, both would be typed as A blood group, while BO and BB both would be typed as B blood group. If A and B both are present, it would be typed as AB because they are codominant. While if both A and B are absent, then it would lead to the blood group be called as O blood group system. So O blood group system is basically, it does not have any new antigen. It is the absence of A or B antigen. The genes for these blood groups are located at 3 different loci. The presence of ABO expression on the red cells is also controlled by a separate gene, which is the H gene. H gene is basically important in order for A antigen and B antigen to latch on to the RBC surface. The H gene encodes for an enzyme. Similarly, the B gene codes for an enzyme. Similarly, the A gene codes for an enzyme. Basically, what these genes do, they code for separate enzymes, which adds separate sugars to the already present precursor substance on the red cell surface. So there is a precursor substance on the red cell surface. The H gene comes into play. It adds a fucose sugar on the red cell surface and makes the H antigen. Then the A and B antigen would come into play and they would add separate antigens on top of the H antigen and make the blood group either B or O. If H antigen is present but A or B are not present, the blood group would be typed as O. So to summarize, what is the type A blood group? A person with type A blood has A antigens on the surface of their red blood cells. They do not make antibodies against A antigen. They make antibodies against B antigen. What is type B blood group? A person who has B antigens on their red cell surface do not make antibodies against B antigen. They make antibodies against A antigen. What is O blood group system? There are neither A antigen nor B antigen present on the red cell surface. They make antibodies both against A antigen and B antigen. Both antibodies are present in their sera. They are known as universal donors because O blood group can be transfused to either a O patient, A patient, B patient, or AB patient. What is type AB blood group? These individuals have both A or B antigen, A and B antigens on their surface. They do not make either anti-A or anti-B in their serum, and they are universal recipients. This is a small chart summarizing the 4 blood group systems that we have seen till now. So this is a type A blood group. It has A antigen on its surface and it forms anti-B in the serum. This is type B blood group red cell, which has anti-B on its antigen B on its surface and anti-A in the serum. This is type AB blood group red cell, which has both antigen A and antigen B on the red cell surface. It has no antibody of anti-A or anti-B type. And this is the O blood group system, which has no antigen of A or B type on the red cell surface, and both anti-A and anti-B are present in the serum. Now, what are the practical aspects of ABO grouping? So, in order to validate a blood group, we have to test for both the antigens and antibodies. They are called as forward grouping and reverse grouping. Forward grouping involves the grouping of red cells to see which antigens are present on the surface, and reverse grouping means testing the serum to see the presence of antibodies. They both should be performed in parallel and they both should be validating each other in order for the blood group to be confirmed. The testing for ABO blood grouping has to be done at room temperature because the antibodies which are formed against the ABO system, they are of IgM type. IgM type antibodies are cold reacting, so they are tested at room temperature. If you test it at 37 degrees, it would weaken the reaction. 37-degree temperature is used for testing IgG type of antibodies. So first of all, there are 3 methods that by which we can test the ABO blood grouping. We'll see all the 3 methods one by one. Labeling is very important while testing for any blood group system. If serum and cell both are to be added, always remember serum has to be added first to the tubes. Results should be observed immediately after the reaction has been performed in case of tube testing, and hemolysis is interpreted as a positive result. Agglutination is also interpreted as a positive result. Then what are the techniques to type for ABO blood groups? First of all, we'll see about the tube technique. We should have clearly labeled samples. They can be plain as well as EDTA. Both are required. EDTA is required to separate the cells for the testing and plain is required to obtain the serum for the testing.
The sample can be stored up for up to 2 days at 4 degrees Celsius and test can be performed. Hemolysis should not be there in the serum or the plasma while you are performing the cells. If the serum has not completely separated from the cells, the tubes should be centrifuged to make the layers separate. And in order to perform testing in the tube technique, you have to make a cell suspension of 2 to 5%. The percentage of cell suspension varies according to the testing method. Conventional tube technique, it has a longer incubation for antigen and antibody, but you can perform that without drying, so that is the advantage. Tubes should be centrifuged to enhance the reaction. It can also detect very weak antigen antibody reaction. Result can be comfortably read as long as the tube is not dry. Cell grouping is called as forward, serum grouping is called as reverse. We have already covered that. So how to perform forward grouping for the cells? You need to take 3 or even 4 tubes for forward grouping. You need to label them as anti-A, anti-B, anti-D, or if 2 separate anti-D sera are available, you need to label them as anti-D1 and anti-D2. As we have read before, serum is to be added first. So in this case, the serum is the commercially available one. So add 1 drop each of anti-A, anti-B, and anti-D or D1, D2, whichever is available. Then add 2 drops of 2 to 5% suspension of test red cells in each tube. Then you have to mix it by shaking gently, leave it at room temperature, and then centrifuge it for 1 to 2 minutes at 1000 RPM. Then you have to take out the tube from the centrifuge, gradually roll it in your 3 fingers against a well-lit background. Disperse the cell button to check for agglutination. If the cell button is getting dispersed, that means the reaction is negative. If the cell button is partially getting dispersed or not getting dispersed at all, that means it is a positive reaction. Now we come to reverse grouping. You have to take 2 test tubes and label them as A and B cell. You have to add patient serum in both the test tubes and add A cell suspension, 2 to 5% A cell suspension to the first tube and 2 to 5% B cell suspension to the second tube. Now you may be thinking, what are these A and B cells? These are the pool cells which are already available either commercially or you can make it in your blood bank. This reverse grouping acts as a confirmation for the forward grouping. Similarly, you have to mix it gently by your hand, let it stay at room temperature for 2 to 5 minutes. Then you have to centrifuge it and check the results by rolling it between your fingers. This is how a working table should look like. You have a basket which contains empty test tubes. Then you have test tube rack, you have anti-sera, you have normal saline, water, distilled water, it should be preferred. Then you have a pipette, a tub for disposing the liquid, the fluids, and the table should be covered by a clean blotting paper. Pipette should be dipped in the normal saline at resting. This is how the red cell suspension looks. So the left tube indicates 2 to 5% suspension. The color should match this. And the right one is a 1% cell suspension. 1% cell suspension is used while testing the blood groups via column agglutination technique. So this color is for 5% cell suspension. Tubes can be labeled as forward grouping, RH grouping, and reverse grouping. We have already covered that. Now, how to interpret forward and reverse grouping? So take the first example. If there is no reaction with anti-A anti-serum, there is no reaction with anti-B anti-serum in forward grouping, but there is reaction in the reverse grouping with both A cells and B cells, then the blood grouping is interpreted as O. We are not talking about RH blood grouping right now. We'll cover it in the upcoming slides. Then the second example is of A blood group. How the reaction would show in the test tubes. In the forward grouping, you would get a reaction with anti-A anti-sera. You would not get a reaction with anti-B anti-sera. There would be no reaction with A cells in the reverse grouping, but there would be a reaction with B cells in the reverse grouping. Next, we come to the microplate method. This is used when the large number of blood groups are to be tested. This can be done manually or this can be done by automated method. It is a very good method for automation. Automation allows for a large number to be tested. It is reproducible. The tests are stored in this computer system for a very long time. There is no manual error or interpreter's error while interpreting the result. So these are certain advantages of microplate method. Then we also have column agglutination technique. You can use column agglutination technique both by manual and automated methods. There is a gel card is there. You see this is a gel card. Gel card is made up of usually 6 columns. Each column has a reaction chamber. Here you see each column has a reaction chamber and a gel column. So the reaction chamber is for when you are putting the cell suspension and serum. Here is where the incubation and the reaction of antigen and antibody happens. Then when you centrifuge this card, this liquid, this test liquid, it gravitates down to the column. If there is an antigen antibody reaction, it is not able to pass through the fine sieve of the gel or the glass bead. But if the reaction is negative, then the red cells, they all settle to the bottom. So here, this is a negative reaction. This is a positive reaction. It is graded as +4 here. Now, what are the problems that you can face while performing ABO blood grouping? There can be clerical errors of labeling the tubes. There can be identification errors that a wrong patient sample has come, or you have picked up a wrong antisera or put the antisera in the wrong tube. There can be improper techniques. The instruments are not clean. The centrifuge is not calibrated. The setup, your tube and your card, they have not been properly incubated. There can also be problems with the newborns and extremely old age people blood group testing, because newborns usually have weak antigen expression as well as weak to no antibody expression. And old people frequently have no antibody expression left in their serum. There can also be problem with certain diseases like multiple myeloma and hemolytic anemias. As we've discussed, dirty glassware, contaminated reagents, centrifugation, all these errors can be there. Now we come to RH blood group system. So there are 5 main antigens in the RH blood group system. The most important one here is D, and the rest, which are called as main. Now we come to RH blood group system. The main antigen here that we test routinely is the D antigen. The other antigens of the RH blood group system are capital C, small c, capital E, and small e. RH blood group system is a very important blood group system because 95% people are RH positive while 5% are RH negative in India. Exposure of RH positive blood in a RH negative individual will frequently lead to antibody formation. Most of the RH antibodies are IgD type. They react at 37 degree. They are not naturally occurring. They are only formed after exposure to RH positive blood. They do not fixed complements, but they are very important when we read about hemolytic disease of fetus and newborn and hemolytic transfusion reactions. Normally, we only type for RHD. If we type for capital C, small c, capital E, and small e, it is called as extended RH typing. It is usually recommended to test D by 2 separate antiseras and label them as D1 and D2. This is a pictorial representation of how the testing is to be done in a tube method. You add 1 drop of anti-D1 to 1 tube, 1 drop of anti-D2 antisera, which is commercially available, to second tube. Then add 2 to 5% red cell suspension, 1 to 2 drops, and 2 to 5% red cell suspension in the second drop of the patient or the donor. Then you mix and centrifuge it, and then you look for agglutination, similarly as you did in the ABO blood grouping. Thank you so much. The errors of blood group typing, they remain the same as the ABO blood grouping.